So good evening. I will start my class now. I'm sharing my screen. Is it visible?
Yes, sir.
Yes, sir.
Okay. So the part is the enzymes used in genetic engineering. So basically, what you do is it's cutting and pasting. That is the first two things which children learn. And the tools which you use are scissors and glues. You cut and then you paste. So basically what you do, you cut DNA with a restriction enzyme, you have sticky ends, and then you paste it in some other place. This is what you do. So similarly, cutting DNA and pasting DNA fragments together typically are among the first techniques learned in molecular biology lab and are fundamental to all recombinant DNA work. Recombinant DNA is DNA from one organism attached to the DNA of another organism. You recombine, cut, paste. So such manipulations of DNA are conducted by a toolkit of enzymes. So what are these enzymes? Restriction endonuclease. They are used as scissors to cut. Then you have DNA ligase. After you cut, you have to paste. And you have other enzymes that modify DNA and that helps this process of cutting and pasting. So DNA modifying enzymes. So restriction enzymes and DNA ligases, they represent cutting and joining functions in DNA manipulation. So apart from this, all other enzymes involved in genetic engineering fall under the broad category of enzymes known as DNA modifying enzymes. And these enzymes are involved in the degradation, synthesis, and alteration of nucleic acid for cutting and pasting, and eventually cloning. So cutting, modifying, and joining DNA, it requires catalytic protein molecules, which are enzymes purified from different types of cells, and they are available from commercial suppliers. So these enzymes, they belong to one group is the restriction endonucleases. That is, they cut DNA at definite sites. They generate DNA fragments, or you can use them for restriction mapping. This right now you don't have to bother yourself, but generating DNA fragments. So DNA modifying enzymes, you have nucleases, polymerases, and enzymes that act on termini or the 5 prime or 3 prime ends. You have nucleases, two types, endonucleases or exonucleases. So endonucleases, they cut DNA inside and from inside, and exonucleases from outside, and endonucleases from inside. Then you have polymerases such as DNA polymerase, DNA replication, clenofragment, reverse transcriptase. These are polymerases. And you have enzymes which act on termini or on the 5 prime 3 prime end. Example, alkaline phosphatase, it removes the 5 prime phosphate. Polynucleotide kinase, it phosphorylates, or terminal transferase, it keeps on adding T towards the end. So these are the DNA modifying enzymes. And you have the DNA ligase, it joins DNA molecules, it forms phosphodiester bonds, and it is used to generate recombinant DNA. Clear?
Sir.
Okay. So let us look at the types of modifying enzymes. First, the nucleases. As the name says, it digests nucleases. Nuclease enzymes that degrade nucleic acid, DNA and RNA, by breaking the phosphodiester bonds that holds the nucleotides together. Nucleotides are joined in a chain, and these by phosphodiester bonds, and these nucleases, they digest it. Then you have the restriction enzymes. You can cut at a definite site. So, and then you have another group of nucleases which degrade DNA from the end of the molecules. and are known as exonuclease. বাইরে থেকে ভেতরে কাটে, আর ভেতর থেকে কাটলে endonucleases. Apart from restriction enzymes, there are four useful nucleases that are used in genetic engineering. One is BAL31, exonuclease 3, DNase 1, S1 nuclease. ঢোকার চেষ্টা করছে। S1 nuclease, DNase 1, exonuclease 3, BAL31. So these enzymes differ in their precise mode of action. How they act, they differ. So mode of enzyme, nuclease BAL31, it is a complex enzyme. Its primary activity is a fast-acting 3' exonuclease, which is coupled with a slow-acting endonuclease. When BAL31 is present in high concentration, these activities effectively shorten DNA molecules from both termini. So, what it does first from the 3' end, it keeps on digesting, cutting, cut, and it also slowly it cuts from minus on a specific site inside BAL31. Exonuclease 3 is a 3' exonuclease that generates molecules with protruding 5' termini. So it digests the 3' ends. Result, the other side has got a protruding 5' end. DNase 1, it cuts either single strand or double stranded DNA at random sites. কাচ করে পুরো ডিএনএ কে উড়িয়ে দেয়। Then you have nuclease S1. It is specific for single stranded RNA or DNA. For single stranded nuclease, S1 nuclease. So BAL31, it is a fast acting 3' exonuclease. It is coupled with a slow acting endonuclease. When BAL31 is present at high concentrations, they shorten the DNA molecules from both termini. And you have seen exonuclease 3, it is a 3' exonuclease that generates molecule with protruding 5' termini. And DNase 1 cuts either single stranded or double stranded DNA at essentially random sites. And you have the nuclease S1. It is specific for single stranded RNA or DNA. Apart from DNases, you have RNases. You can get rid of RNA with this. RNases are everywhere. So moment you take RNA, touch it with your hand, that RNase is going to destroy that RNA. Polymerases, you know, I've heard about polymerases. They synthesize copies of nucleic acid molecules. You have heard about DNA polymerase. So you have a DNA dependent DNA polymerase and an RNA dependent DNA polymerase and a DNA dependent RNA polymerase. So DNA copies DNA into DNA. RNA dependent DNA polymerase copies RNA into a DNA, and DNA dependent RNA polymerase, it transcribes DNA into RNA. RNA polymerase যেটা. These enzymes, they synthesize nucleic acids by joining together nucleotides whose bases are complementary to the template strand bases. So one strand on the other side, the complementary strand is synthesized. The polymerase works like this. Synthesis, as you know, it proceeds in the 5' to 3' direction and requires a free 3' OH group for the formation of phosphodiester bond. Remember DNA replication. And you require a primer also with an exposed 3' OH. Otherwise, they don't work. Polymerases are the copying enzymes of the cells. They are template dependent either on DNA or on RNA. DNA polymerase 1, in addition to the polymerase function that is synthesized in 5' to 3' direction, you have 5' to 3' and 3' to 5' exonuclease activities. So DNA polymerase 1, it has got 5' to 3' and 3' to 5' exonuclease activities. So if there is a mismatch error, this exonuclease will cut it and a proper match will come. Against A, opposite of A, there should be T. By mistake, if G comes, this exonuclease will come and cut and remove, and again the proper T will come. This is the function. A major use is nick translation. So nick translation means you cut with the exonuclease, you put a radiolabeled nucleotide and then give DNA polymerase. This radiolabeled nucleotide is incorporated and your DNA becomes radioactive. You can use it for further detection. Nick translation or head translation was developed by Rigby and Paulberg. It is a tagging technique in molecular biology in which DNA polymerase is used to replace some of the nucleotides of a DNA sequence with their radiolabeled analogs, creating a tagged DNA sequence, which can be used in fluorescent or radioactive southern hybridization or blotting techniques. It is also used for radiolabeling. So you make a nick and with polymerase you fill it up. The 5' to 3' exonuclease function of DNA polymerase 1, it can be removed by cleaving the enzymes to produce what is known as clenofragments. So you can remove the 5' to 3' fragment exonuclease from the DNA polymerase 1, and that what is remaining after removing the 5' to 3' is the clenofragment. This clenofragment has got the 3' to 5' পেছন থেকে কেটে বাদ দিয়ে দিতে পারে exonuclease, and it has got polymerase activity. But this 5' 3' ওটাকে উইয়ে দেওয়া যায়. A clenofragment is basically used. The clenofragment is used where a single-stranded DNA molecule needs to be copied. Because the 5' exonuclease function is missing, the enzyme cannot degrade the non-template strand of dsDNA during synthesis. কারণ 5' 3' নয়তো ডিগ্রেড করে দেবে তো 5' সাইডটা, সেটাকে করতে দেয় না. Therefore, the large or clenofragment of DNA polymerase 1 has DNA polymerase and 3' 5' exonuclease activity. মানে 3' থেকে 5' ডাইরেকশনে নিউক্লিওটাইড বের করে দিতে পারে. It is widely used in molecular biology. So this is the whole DNA polymerase 1, 5' 3' exonuclease domain, 3' 5' exonuclease domain and the polymerase domain. এটাকে proteolysis করে এই large fragmentটাকে is kept and this small fragment 5' 3' exonuclease fragment is removed. This is used in molecular biology, this clenofragment. So you know DNA polymerase 1, it is a template dependent DNA polymerase. It catalyzes the 5' to 3' synthesis of DNA. This enzyme also exhibits 5' the 3' to 5' exonuclease activity. It is called proofreading. Like I told you, opposite to A, if by mistake instead of T there comes a G, then this proofreading, what it does, this proofreading part from the 3' end, kachak say it cuts it off, and the right nucleotide is again synthesized on top of it. So this when this DNA polymerase 1 copies, it checks whether there is one error, means one mutation. It can be catastrophic. So there is a proofreading, and because of this, the errors don't happen. There is a high fidelity. So the clenofragment, you know, the 3' 5' part is removed. 5' 3' exonuclease is removed. So this is a template DNA. You make a small complementary primer opposite chotto primer. You add clenofragment. and this clenofragment, and you give this labeled nucleotide DATP, DCTP, DGTP mixture and clenofragment. It will synthesize in the 5 prime to 3 prime direction. You will get a fragment. Then you have T4 DNA polymerase. It is a viral DNA polymerase. It's a template DNA. It catalyzes 5 prime synthesis from primed single stranded RNA. This has, you can use this also. Instead of Cleno, you can use this 3 prime 5 prime exonuclease activity. It does not have 5 prime 3 prime exonuclease activity. You can use this also. It has got a stronger 3 prime to 5 prime exonuclease activity. That is, it has got a greater proofreading function. It is used to blunt ends of DNA, fill of 5 prime overhangs. For example, if after a restriction digestion you have a sticky end coming out, this will, the polymerase will, what will come, this 5 prime side which is overhang, from the 3 prime side it will synthesize and it will cover it up. It will remove the overhang. And if you give radiolabeled nucleotide, this overhang, the synthesis, which the complementary synthesis which will take place, there you will have radiolabeled nucleotides being incorporated. The T7 DNA polymerase is also another DNA polymerase used. It catalyzes DNA in the 5 prime to 3 prime direction. It's the same thing. It also has got a 3 prime to 5 prime exonuclease activity. It has got 1000 times greater exonucleus than clenofragment. Then you have terminal transferase, TDT. It is template independent DNA polymerase. It catalyzes the repetitive addition of deoxyribonucleotide to the 3 prime OH of oligoribonucleotides and single stranded and double stranded DNA. For this, you don't need a template on the other side. Directly, randomly, it will keep on adding T. At least it needs 3 nucleotides, which has a primer, and behind it, it will randomly, it will keep on adding your nucleotides. Then reverse transcriptase. This will convert RNA into DNA, that is known as cDNA. It has no exonuclease activity. Basically, it is used in RT-PCR. From RNA, messenger RNA, you make cDNA using reverse transcriptase. From RNA, you make DNA. And on this DNA, you can do, on cDNA, you can do PCR. You can generate cDNA, complementary DNA. It complementary to the RNA, this DNA is made. So let us stop today over here. Tomorrow we will talk about alkaline phosphatase, polynucleotide kinase, T4 polynucleotide kinase. ঠিক আছে?
Okay, sir.
Yes.
Okay. তাহলে আজকে এখানে শেষ করছি। এনি কোয়েশ্চেন্স?
No, sir.
ওকে, ওকে।